tenfold that number. And then, of course, if we start talking about xenobiotics, gut microbiome, dietary components, environmental toxicants, we're up into the hundreds of thousands of molecules. Plant metabolites are, the metabolome is much more complex. There's approximately 125,000 known plant metabolites at this point in time. So the next question is, why bother with metabolites and metabolomics? Well, if you want to sort of argue the case, they're downstream of the genome and proteome and more closer to the phenotype. Very subtle changes in RNA or protein levels can have big changes in metabolism. Again, we can sort of argue that the genes and the proteins are just predictive of what is happening at the physiologic level, and so metabolomics provides a snapshot of that. Okay, in the case of the metabolome, as I said, there's a vast number of uncharacterized metabolites, and the type of approaches we use, which is primarily mass spectrometry, allows us to assess these even when we don't know exactly what they are. Okay, so what is new in the field, and why has this field sort of taken off, and why have I sort of moved some of our efforts from our genomic and molecular biology approaches to metabolomics? Well, as far as just giving an overview of the methods, you first have to separate the metabolites by gas or liquid chromatography typically. The LC approach has some advantages, and typically one can assess a larger number of metabolites. The big change in the technology has been the detection. Jeremy Nicholson at Imperial, who really is the father of this field, originally used an NMR approach, which is powerful but less sensitive than mass spectrometry. So these time-of-flight mass specs started coming online, and I've listed here the number of publications where metabolomics or metabonomics is mentioned. And these machines started to come online about 2005, 2006. As you can see, there's just been an explosion, almost an exponential increase in the number of PubMed publications each year since then. So there's now over 6,000 publications in the literature. So why are these machines important? I've given some reasons. It's high resolution, you have high sensitivity, you get exact mass determination. But most importantly, I think, is that it's higher resolution than the original NMR. Typically 50 to 100 metabolites can be seen by NMR, whereas with the modern QTOF approach, 5 to 20,000 peaks in a particular experiment is typical. Also, it is sort of dilute and shoot, particularly for biofluids. You crash out the proteins and you can run, we've run up to 1,500 samples at a time. Finally, and this was supposed to appear, and now I guess it will appear, we have been designated Water Center of Innovation in Metabolomics at Georgetown, so we're the first in the US. Frank Gonzalez at NCI and several others also have centers now. And from a funding standpoint, this has been beneficial for many of the faculty at Georgetown. Over 40 are using the facility actively. We have over 20 outside users. We're now part of a Mid-Atlantic consortium where we're providing the metabolomics support for universities all the way from Hopkins down south to the University of Virginia. And for those of you who know about the NIH grant system, there's a variety of grants that our metabolomics is providing an important component. So these are the types of machines we use, and so we're a Waters shop, and we're using the Waters machines primarily. And what we get here is a continuous spectrum. So with time here, the peaks will be coming out with time, and you can measure the particular mass and the intensity, or you can plot it versus mass and elution time from the chromatography, the liquid chromatography output. And the take-home message is that we're getting this continuous three-dimensional type of plot with time. A typical experiment can easily generate DVD size 3 to 5 gigabytes of data. The typical workflow is we get the chromatogram, and let's say we're comparing normal versus malignant. We identify particular peaks that show a difference between, let's say, control and cancer. They are then put through a variety of statistical packages. When we've identified the ones that we think are important, we will use purified standards to validate by MSMS and MRM quantification and the like. So there's a heavy analytic chemistry component to this. So just to mention, before going into the cancer metabolomics, what we have found doing this is that the computational challenges are immense, and it's really sort of, using American term, the Wild West as far as how you want to analyze the data. There have been a variety of groups making important contributions, such as Gary Shustek at the Scripps. And some of the problems that we're faced with is that it's somewhat dissimilar from some of the typical data sets, such as one sees with microarrays or sequencing. Some of the problems are very high noise levels, particularly with human populations. One thing that drives the statistic people absolutely crazy is missing data, because you're doing peak alignment, and so zero filling is problematic when you're trying to assess the importance. And then, of course, biofluids, there's issues about normalization and the like. And then finally, there's a staggering yield of quantitative data that requires to be considered. So what we have done is we've put together a variety of different statistical packages, which I won't go into here. Some of them are relatively simple, such as this VAMP package, which allows sort of a graphic representation of the changes in particular metabolites. And we've developed others which are in the process of being published, such as SPICA, which is a novel approach that doesn't measure the actual levels of a particular metabolite, but compares metabolite pairs. So the advantage there is you actually technically do not need a control sample, so you're doing internal normalization. The most important, I think, component from a day-to-day standpoint is this metabolizer package, which Titus Mack in my group with Leah Leaki and others in my group have put together. And so this is a comprehensive package that provides statistical support and then allows interfacing with some of the small molecule databases to identify or get putative identification of metabolites from the peaks, and then also go on to pathway analysis. So the importance of metabolizer is that many of the biologists trying to get into metabolomics, it's just a daunting problem, just trying to get started, and many of the packages are not particularly user-friendly. So this package has been made to be user-friendly, and it's commonly used in my lab and others at Georgetown, particularly for novices in the field, those who are new to the field, but also for those who've been in it for some time. So some of the output is listed here. We can do PCA and MDS type plots for statistical significance, heat maps, correlation analysis, KEGG pathway integration, and a variety of other sort of graphic displays of the data. And so a particular workflow is here. There's an interesting zero filling approach, filtering approach rather than a zero filling approach, which was actually quite appealing to the reviewers who reviewed this for analytic chemistry recently. And so this paper is now in press, and you should be able to get a copy soon, and download the software from our site and get working on it if you wish. As far as identifying particular metabolites, this particular software package will access some of the major databases that are out there and give you putative identity and also give you pathway analyses before you go on to identify particular metabolites, specifically using both, using targeted approaches of various sorts. Okay, in the case of cancer, we know that tumor suppressors and oncogenes affect metabolism. I just show a very simple schema here. For my friend P53, which my group has worked on for many years, we know that this is a master transcription factor, which is a tumor suppressor, And it controls a variety of genes that are involved in metabolism. I've shown several here, such as Tiger, SCO2, and the like. So these genes are directly impacting metabolism. AM kinase feeds into P53. AKT has a pro-growth and anabolic effects, and is also associated with cancer signaling. And so when these genes are overexpressed, or in the case of P53 inactivated, you get a substantial change in the metabolism of the cell, and some of this has been out by a variety of groups over the years. And just to show that we're not the only ones interested in cancer metabolomics, here's another plot of the number of PubMed publications that come out each year mentioning metabolomics and cancer together, and there's almost 1,000 out there now. So this is an area of active investigation. Now, as Pablo mentioned, our laboratory's major focus and much of our support is on stress signaling and injury responses. So the idea here is that I was going to try to leverage Jerry's talk on cancer genomics to show how we can expand on that with metabolomics. Okay, so what is our ultimate hope? Well, our ultimate goal is to be as good as a dog. And what I show here is a report from a Japanese group that trained a particular bomb-sniffing dog and they reported that they had up to 98% accuracy in predicting cancer in early cancer detection by having the dog smell people's breath. So the sensitivity of the dog olfactory system is greater than any of our instruments, but that's a goal that we're shooting for. Okay, in the case of P53, I'll just mention briefly sort of the massive changes that we see. Here's a PCA plot where we run a variety of independent isolates of a cell line, which is a P53 wild-type line, TK6. And the proximity of the points is how similar the metabolomic spectrum is, and the TK6 you can see clusters, and in a subline where the P53 has been knocked out by gene targeting, you see a clear separation. You can also see this in wild-type and P53 knockout mice, and what Leah Leaki in my lab has done, we're showing here results with urine, is we've looked at changes in metabolites with red going up, and here for particular metabolite peaks, which are changing after ionizing radiation, which will turn on P53 and other tumor suppressor and oncogene-related pathways. And what we see here is a substantial difference in this heat map for the P53 knockout mice versus the P53 wild-type mice. And again, this highlights the striking differences between normal and malignant cells. So the question that we want to ask is, can we take advantage of these differences to start developing approaches where we can perhaps see some changes in easily accessible biofluids rather than having biopsy patients and possibly in the long term being used to monitor for patients who are at risk. And so what we decided to do was to use a well-characterized mouse model for a pancreatic adenocarcinoma. So there's a particular oncogenic RAS which is activated when crossed into a Cree background, and these mice develop a sort of a predictable progression from pre-malignant lesions to frank adenocarcinoma with some similarities to human pancreatic cancer. And so the different types of pathologic changes are shown on this slide. So we have these intraepithelial neoplasias, or PAN-II, which then progress to frank PDAC, or a pancreatic adenocarcinoma. And the variety of additional mutations occur as the tumor progresses. So the question that we wanted to ask was, Leah wanted to ask, was what can we see at the metabolite level? And this just highlights that this approach, when the animals have the activated transgene, oncogene in them, is that we get these lower grade tumors at earlier times, and then at later times we can go all the way on to frank adenocarcinoma. So we can compare mice with the transgene who have different diagnoses when they're sacrificed, as well as look at the control animals where the transgene has not been activated. And what we see here is that we focused primarily on serum metabolite analysis. We also carried out urine metabolite analysis, as well as assessing changes in the tumor per se. And if we look at the serum metabolite analysis, what we see here for the control, I'm trying to use this mouse here and it keeps advancing. But what we see here is that on the left we have the control mice. On the right we have the mice that are starting to develop these early lesions. This is a heat map, with red being increasing expression of metabolites, and you can see a clear difference with each column being an individual mouse. By the time we get to 13 to 16 months, you can see an even more pronounced difference. So just as sort of a proof of principle, Leah in my lab went on to identify a few metabolites, and what I'll show you is some results here for a particular metabolite, namely citrate, where she found an increase that was statistically significant for the adenocarcinoma, particularly at the later times, and we saw the progressive increase from wild type to these early intraepithelial lesions all the way through to adenocarcinoma. And the interesting finding was that when Joe LeConte and Anton Wellstein's lab went back and screened for citrate synthase in the actual tumor, and the tumors also showed an increased citrate. What he found in the staining here is by immunohistochemistry is we find staining with the tumor but not in the normal tissue. So we're able to work back and look at the particular enzyme responsible for this, which is upregulated in the tissue. And so citrate is probably not the ideal metabolite, but it does sort of show a proof of principle that we can work back from the small molecule back to the enzyme. Okay, so the conclusions there is that we were able to detect the metabolite in the circulation. This is the first report of it in the serum. There have been some studies in humans showing increased citrate synthase activity in human pancreatic cancer. So you can say, well, this makes it less original, but I think it also gives us confidence that this model has some relation to what one would expect to see in human cancer. So there's ongoing studies that are going on now to assess additional metabolomic changes, and I guess what I'd ask you to do is to stay tuned and also to correlate this with patient data sets. So in the last few remaining minutes, I just wanted to mention some work from Marina Cheeman in my laboratory, who has her own shop, and she's co-director of our core. And this is an American Cancer Society pilot project that she carried out. And this was a tissue and matched serum samples that were obtained from the DiviMed repository. This is a relatively limited number of samples, and the numbers are listed here in this slide. And what we have found is that the surgeons and the pathologists, and me being a pathologist, I guess I should take partial responsibility, is that when a tissue is excised, our highest priority is not getting it into liquid nitrogen quickly. And part of the problem is that sample collection is critical. If the tumor sits, for example, in the operating room suite for 5, 10, 15 minutes, it gets hypoxic, there's a variety of metabolic changes, and the metabolism is going to change substantially. And this is a real problem with metabolomics. Now, DiviMed, which is a company, has developed, and in collaboration with Georgetown, a sort of a very customized, rapid collection of tumor samples and rapid freezing. And that's really proven to be critical to get a reliable signal. And Rita also used non-negative matrix factorization and scree plots to try to group the different spectrum of metabolites that she found. And what she found is that, and was that there was some, particularly with the adenocarcinoma, pancreatic adenocarcinoma, is that there was dysregulation of lipid and nucleotide metabolism-related pathways. And so she's comparing pancreatitis, the inflammatory disease, where sometimes there's surgical specimens, to patients with these early neoplastic lesions, which are not particularly frequent in surgical pathology, but there are a few that can be identified. And then a pancreatic adenocarcinoma, and then finally colorectal adenocarcinoma with unrelated cancer, which was used as a control group. And so she worked with the tissue and then expanded it to the serum. And this is the flow chart that is being used. It's sort of a typical flow chart. You can modify the columns as well as the solvents and the approach you use to assess for both a wide variety of metabolites, as well as for looking at lipid metabolites. And so this is a sub-area of metabolomics referred to as lipidomics. And the take-home message here in this heat map is that she identified 7 scree sort of clustered groups, and when she analyzed these, the adenocarcinoma, which is C7 on the left side of the screen, shows distinguishable differences from these early ductal changes, that is the IPMN, and that differed from pancreatitis. And then the colorectal, there were several scree groups that indicated some diversity in these tumors. And so this was sort of an encouraging first step in this study. And what I'd like to do in the final minute or two is to say, well, how do we hope to use this in the clinic? And Subha Madhavian was brought in from the NCI a few years ago and has put together a program referred to as the Georgetown Database of Cancer. When you're putting together these initiatives, it's important to have a nice initial, so it's called GDOC. She provides t-shirts to everyone at Georgetown, so you'll see that if you come there, people wearing GDOC t-shirts. Here it comes. And so the way she's built this is that we have a large cancer catchment, which is the bottom of this pyramid. The next is the clinical and epidemiologic studies. And the idea here is to incorporate that with gene expression, DNA analysis, and as well as proteome and metabolome and kinome analyses. So all of these are being incorporated into a master database and integrated. And so her job is the Georgetown Database of Cancer, which is the orange block, and then that will go to prognosis, biomarker, and drug development. And finally, the idea at the top is to have a systems medicine and clinical approach. So the doctor of the future will sit down with gigabytes of data, and will use these types of tools to inquire on the genome, the metabolome, the proteome, and all the clinical parameters that are available. And with the cost of DNA sequencing and the like coming down, those days are really fast approaching. So in this example here, I think I'm going to diverge a bit from pancreatic cancer and look at the IndiviMed 40 patient set that we had for colorectal cancer. And so here, is what she did was carried out a genome-wide profiling for 40 early stage 1 and stage 2 colorectal cancer patients. And so with the IndiviMed, what we did is the samples were frozen, and then they were assessed 2 years later, and we looked at the results and asked which of the patients failed, namely which of the patients developed metastatic or recurrent disease compared to those who should have been cured by the surgery alone, since it was localized cancer. And so the idea then was to identify molecular signatures to serve as prognostic markers of recurrence, and then to optimize this for chemotherapy, presumably in the future. And so the key abnormalities were in genes, gene expression, metabolites, DNA copy number, and the like. And what was interesting is when we did the analysis, we could distinguish the people who were the winners versus the losers at 2 years. And the most frequent and predictive, interestingly, were at the metabolomics level. And I should also mention that by a donation by the Rush family, we have a Rush Center for GI Cancer Research, which is part of the Lombardi Comprehensive Cancer Center at Georgetown. And so this is the matrix that Suba and her team put together, and Amrita and our team in the genomics, in the metabolomics score, carried out both urine and serum metabolite analysis of the patients at the time of surgery. And so we have miRNA, gene, copy number, transcriptomics and the like, and these different colored boxes refer to that. There's a variety of filtering approaches, and then analysis of top omic features of the relapse on the left here versus the various features which can distinguish these two groups. So this paper has been recently published in one of the genomic journals, and I'll refer you there for the specifics. But the take-home message here is that there was 31 metabolites, don't seem to be able to use the pointer very well, that strongly associated the CRC relapse, and these 31 markers pointed to pathways related to immune response, immune cell signaling, and trafficking as predictors of relapse risk. And if we look on the right, this sort of a star-shaped system here, Suba has and Amrita have color-coded the different markers. So the red are upregulated metabolites, the brown are downregulated metabolites. And as you can see here, and this is from serum and urine, not from the tumor per se, although they did work back from the tumor as well, is what we can see here is that the majority of the metabolites that gave a distinguishing separation between the relapse-free and the relapsed cases were in fact metabolites. There were others such as gene expression and copy number, but the number of particular markers was less. And then down at the bottom, Suba has used her system's magic to put this together into several different pathways involved in inflammation and infection and the like. So that is sort of the hope that we have, is to be able to integrate all of these different omic approaches and to use this to make smarter diagnoses about which patients have a tumor where the biology is going to indicate early metastatic spread, even if the surgeon doesn't and the pathologist don't see it, and as well as therapy in the future. The most important slide here is the contributors, and this is the way Georgetown looked a few weeks ago. We still have some foliage on the Potomac, and Leah and Titus are in my lab, and then Anton's lab is nearby with Joe LeConte, who's now actually a medical intern, and Amrita Chima and her staff were also key players in the metabolomics operation. And then the GTOC team, led by Subhab and Havian, is a large collection of talented individuals, which I've listed here. So hopefully I've given you an overview of modern metabolomics and what you can do with it, and how even with these limited pilot projects and using the mouse model as a guide, I think there's good promise then that both in serum and urine and probably other easily accessible biofluids or breath metabolomics, we are on our way to developing additional biomarkers that will help clinical care. So thank you for your attention, and I guess I can take questions if you wish.
Thank you. Thank you. Grab the mic. Thank you, Dr. Farnak. Could I ask the first question? Is the mic working? Huh? Yes. Thank you for your elaborate lecture. As a surgeon, could I ask you the first question? Pancreatic adenocarcinoma is a bad disease, and And actually it's one of the cancers which give cancer a bad name, because most of the patients, they die in 2 years. Whatever you do, you operate, you don't operate, you give this, you give that. So the way I understand it as a surgeon, as a clinician, with all this metabolics, genomes, metabolites, I need you to give me something to diagnose those patients as early as possible, because this is what is the cure. So what's your vision in that?
Well, I think in the case of pancreatic cancer, it is a horrible and oftentimes hopeless disease. There was one of the members, I think, of the Supreme Court, a woman, who actually had an MRI for unrelated reasons, and they picked up an early pancreatic cancer that was cured. So it is curable, as you know, and the idea here is to develop biomarkers that can presumably detect it at an early age or an early stage. And I think the beauty of metabolomics is that, yes, these machines are expensive and difficult to use, but once you identify a subset of metabolites, whether it be a few or a few dozen, that's really just clinical chemistry. And so that's something that could be done in a screening way as people, if you want to assess patients at risk or even with age. And I think that there's one slide that I show sometimes of a character from a TV show called Star Trek. Mr. Spock had a tricorder that would record, and you pass it over a person and he could sort of record all of their biology. And I think that's sort of a focus of many of the NIH institutes, both NIEHS and NCI and the like, to develop some of these biomarkers that can be assessed early on.
Well, what's the answer to my question? Are we getting there, or what we are doing?
Well, I think at this point we're in the process of characterizing the biomarkers, and I think there's promise, but as far as commercialization, there has been some, as you know, for certain types of breast and lung cancer and the like. But I think that these will be coming in the next 5 years, and I think that I'm optimistic that these types of approaches, particularly as the costs come down, can be used for screening.
Thank you for the really interesting talk. So I was really interested in seeing that you have, at least in the case of P53, some type of signature that appears to change dramatically the metabolomic profile. Do you think that there is some type of a parallel between different type of tumor suppressors and how different type of tumor suppressor might induce different type of metabolomic signatures? And you can imagine the same point for oncogenes, but I'm really interested in the tumor suppressor aspect.
I think that the underlying molecular defects are going to affect the metabolism. And I think that's part of the problem until recently. I've been on some of these metabolomic study sessions, and the people who are absolutely superb at the chemistry don't know much about the biology. And I remember one person giving a fantastic review to a grant with a few non-isogenic lines where we had no idea what pathways were perturbed, and they were all enthusiastic about that. So I think that this is an area that I think is needs to be characterized, and I think the tools are there. It can be done relatively quickly by whoever wants to, but I would predict, based on just the simple things I showed with P53, that there will be a big change. I'd also refer you to the NCI 60. There's a fair number of omic studies in that cell set, including metabolomics now, and there's a variety of interesting features that have come out. Of course, these lines have hundreds of different mutations, but you can see some clear P53 signatures, for example.
So I think maybe we have some time for questions from here. Yeah. Okay. I see two hands. One there, one over here.
So here's a scientific question. I always had a problem with is, metabolomics type of approaches, because they really left me asking the question whether we're really seeing the cause of the disease or the effect of the disease. And can we really find out cooperative effects? I think what people like Benjamin Cravat at Scripps are doing, looking at functional metabolomics, and I think one of his postdocs now at Harvard, the same. Do you think that should be a bigger priority, or what's your take on this?
I think that from a biomarker standpoint, it doesn't matter. If the biomarker is there and you can use it for early diagnosis or to make predictions on clinical outcome, it doesn't matter. I think that what you say is correct. I think the only way to address these types of issues is a genetic approach at the current time. And for example, using a mouse model approach or a knockdown approach in cell lines and the like, and knock out individual pathways and start sorting this out. I think that one of the advantages of metabolomics, I thought you were going to criticize the fact that we don't know many of the peaks are, but sometimes you'll find a peak that's not in the database. For example, Gary Patty found a particular ceramide-related compound that he published in one of the Nature, I think, Chemical Biology a year or two ago that was involved in pain mediation in the spinal cord. And he found a particular peak, which was not actually known at the time, and then went back and did the fragmentation analysis and eventually, uncovered the particular metabolite. So I think that not knowing what the peak is sometimes is an advantage when you're doing these survey type experiments. This is really pure untargeted metabolomics when you're using the QTOF approach. There are targeted metabolomics, which I think one of the other speakers at least will be speaking about.
Okay, then I'll leave my second question.
Yeah, thank you, Doctor. Thank you. I would like to thank also the Center of Excellence for organizing this international conference. My question here, as the doctor said, the pancreatic cancer is one of the worst diseases. It's a bad disease, as we know. And most of the patients would die during the 2 years from the diagnosis. This is the problem with the detection of the disease. Most of the patients, the disease was diagnosed at the late stage. So we have a problem with the detection of the disease.
We go to the question.
The question is, do you think, Doctor, when we know these biomarkers, if we target with some molecules or some molecules or some compounds or some chemical molecules, if it's a curative way or it's a good strategy to at least to prevent the disease, for example. If we know that the metabolites or the cellular metabolites, if we target these chains or these biomarkers, we can prevent the disease. We can prevent the cures of disease at that point. Thank you.
I think it could be used to identify patients or individuals at risk, and I think there is a lot of epidemiology. One thing I should mention is the cost of doing the high throughput LCMS, at least when you have your own shop, it's about 10 times cheaper than running, for example, the RNA arrays. So you can do a lot of samples. We've run up to 1,500 sample sets. I think that the point of showing the pancreatic mouse model is that these intraductal dysplasias that did give metabolomic signature both in the pancreas as well as in the serum, these would be akin to an easily resectable tumor in a patient. So the idea would be is not everyone can have yearly MRIs or MRIs every 6 months and CAT scans and the like, but if we could identify subsets of people where suspicious metabolites, which were screened much more cheaply, then they could be brought in for hopefully early intervention. I think the question of at-risk populations and the like is complicated, and there's a tremendous amount of effort in the field already on that. And you'll hear about some of that later today.
I have a question.
Go ahead. Yes.
Yeah.
Fine question.
Okay, thank you for this presentation. I have actually two parts to my question. The first one, the sample preservation. We know that the metabolism change very quick within the sample and it get quickly hypoxic before get examined. The optimum time and how we can be sure that the chemistry within the cell is not changed any time processing till we examine it. The second part is the citrate synthesis. The citrate synthesis get affected. Actually, the acetyl coenzyme A control it. Just I want to know the effect of the hormones insulin and glucagon as controlling the metabolism of lipids and carbohydrates in this process. Thank you.
I did not get the second part, but the first part is about how we optimize getting the specimens from the OR. And this is as a surgeon, you know, we have that problem every day. The people in here, they want fresh specimens. The pathologist, he tells me, don't even touch the specimen, send it to me. And there is logistic problems in here. People in the lab, they have to come with innovations, you know, how to keep the specimens fresh from time zero. This is the question. Go ahead.
Well, we know that patients do very badly with 4 minutes with no oxygen. And I think that tissue does badly for 4 minutes with no oxygen. So my understanding of the indivity net is that the sample is excised and a portion of it goes into liquid nitrogen or onto dry ice almost immediately within the OR. And so I think that's one of the issues with looking at metabolomics of the tissue. That was done in that was done in the pancreatic study from Amrita Chima that I showed. But she also then went on to validate some of those findings in serum. Now, serum and urine is a different story. There, the collection is easy. It's easy to spin out the serum. Whatever is in the serum is reasonably stable, so you can spin out the serum and freeze it within 5 or 10 minutes. But I think that to isolate for the patient, you really need to change the surgical procedure. And as a pathologist I remember when I was in training many years ago, as they were always very emphatic about looking at margins, I don't know how the pathologists and surgical surgeons are going to feel about a fraction of the tumor going into liquid nitrogen immediately. But that's basically what needs to be done.
Thank you. Maybe you can ask the question, if you don't mind, in the coffee break.
Yeah, we can discuss at the coffee break. There's an echo in here, so I'm having some trouble hearing.
Sure.
Thank you.
So, thank you, everybody. I think we'll go to a coffee break. It's 11 now. We meet 11:15. Thank you, everybody.
I would just like to thank our chairpersons. Thank you very much. And hopefully we'll be back within 5 minutes. See you for the next session. The idea of the conference is that you have interactive sessions parallel to the main talks. So please, avail this opportunity. Please proceed to the stage.
Okay, so we start, and hopefully the rest of the people networking outside will join us. So welcome to the second session of this conference and of today. Unfortunately, the second co-chair is, as you heard, is in this virus attack, so he preferred not to be sort of sharing that coffee with you. So I will lead I will chair alone that session. The first speaker is Professor Ángel Carcedo from Santiago de Compostela. He's one of the leaders in Europe and in the world on clinical genetics, and he has actually very strong activity both on the clinical side as well as the research side. And I had the pleasure of collaborating with him in a research European project that is actually finishing at the end of this year. So he will talk about colorectal cancer treatments and pharmacogenomics. Angel.
Thank you so much, Manolis, and good morning, everyone. I'm going to talk about pharmacogenomics of colorectal carcinoma. It's a subject that I have devoted to this subject my research during the last 5 years. And it's because this cancer is really important, because it's the most frequent in the developed countries. In Spain, it's the one with more new cases per year, and it's the same is true for most of the countries all over the world. It's the second in mortality, and in Saudi Arabia, the frequency of this cancer is increasing very, very quickly. In the treatment for colorectal carcinoma is always the same as for most of cancers, is surgery and chemotherapy. And the survival has improved quite a lot during the last 10 years, especially because of advances in chemotherapy. The introduction of 5-fluorouracil and capecitabine, that is a precursor of 5-fluorouracil, was key, and especially the combination with 5-fluorouracil with other drugs, such as irinotecan in a complex called FOLFIRI, and oxaliplatin in a combination called FOLFOX. With this combination, the survival increased a lot, and nowadays the survival combining surgery and chemotherapy is a little bit more than 50%. That is quite a lot. And this is more or less an average in medicine. I mean, the efficacy of a drug treatment on average is 50%. And it changes a lot from one group to another. And obviously, it's not homogeneous in groups of diseases and treatments, and obviously none in individuals. But the problem of drug treatment is not only the efficacy, but the number of ADRs. This is the adversary drug reactions that we have, that represents 5% of all hospital admissions, is the 4th cause of death in the States and the 6th in Europe. And it represents not only death, but an incredible amount of investment in care. And the reason for this is that it's very much related to the paradox of the modern drug development. When we introduce a new drug, for instance, a new chemotherapeutic agent, we use clinical trials, but clinical trials provide evidence of efficacy and safety in populations. And then the physicians treat individuals, patients individually, and they can vary widely in their response to drug therapy. And I like very much this sentence of this statement of Sir William Osler, because it's very true. The reason for this different response of different individuals is the variability. And the variability is part of life, is the law of life. And he said that as no two faces are the same, no two bodies are alike, and no two individuals react alike and behave alike, and the abnormal conditions which we know as disease. And it's true that we have an incredible variation. And the goal of pharmacogenomics is to study the variations, and that means to identify biomarkers at a DNA and an RNA level related to drug response. And the variations in population is huge. For instance, in enzyme metabolizers genes, the differences among populations are extremely big. Imagine this CYP26 variation, polymorphisms. Here in this part of the world is extremely high. The Mozambiques have only for this gene, 40% of ultra-rapid metabolizers. It's extremely high as well in Saudi Arabia and this part of the world. Not as high, but very high. And only this gene is metabolizing more or less more than 20% of the drugs we are using nowadays. And that has to be with efficacy. And I am very happy to see that in the posters there are at least 3 or 4 posters dealing with seed variation related with different responses to drugs. But the response to drugs depends on 2 different things. First depends on the disease, and second depends on the drug in a given individual. The disease is very, very heterogeneous. It doesn't exist any single disease. There are always lots of diseases integrated in a single entity. It doesn't exist a single colorectal carcinoma. There are lots, and the same is true for any single disease. And the reason of the lack of efficacy is mainly that we think that we treat all the disease as a whole, but there are many, many different genetical causes of the disease and different groups of the disease. And the same is true with a drug in an individual. We have variations in metabolic genes, in transporters, in drug receptors, and all these type of things interact with the environment, with the environment. Because both the complexity of the disease and the drug in an individual are what we call, from the genetic point of view, complex traits. That means traits that they have genetic factors and environmental factors at the same time. In colorectal carcinoma related with the complexity of the disease, and the same as many other human diseases, we have a group that is of cancer, of colorectal cancer, that is very Mendelian, with a clear pattern of inheritance. We have some familial aggregation that is not clearly inherited, but it has some aggregation, and we have many, many sporadic cases. Most of the genes involved in the Mendelian colorectal cancer have been found so far. And thanks to the next generation sequencing technology, most of this variation we have been able to identify most of this variation. But this variation only accounts for 5% of the 35% of the total of variation. I mean, the heritability of colorectal carcinoma is 35%, and the Mendelian form represents only 5% of this total. And we understand almost all the Mendelian forms of colorectal carcinoma. The problem is here, the 30% that has to be discovered. And we have Mendelian forms. To identify the genes involved, we use linkage analysis. But when the pattern of inheritance is not so clear, we have to use association studies. Association studies means to get a number of cases, a number of controls, and to study a number of genetic markers. We use, usually we analyze SNP, single nucleotide polymorphisms, to try to identify the genes involved through the association with a given SNP. And there are 2 types of association studies. One is, we can use 2 different approaches. One is to use the candidate gene approach. For this we need to know, to have candidate genes and to select SNPs in candidate genes. And the second approach is to interrogate the whole genome. That's what we call a genome-wide association study, or GWAS. Candidate gene approaches have been used for identifying genes involved in colorectal carcinoma, but very, very unsuccessfully. Very, very few genes have been found using this type of approach. And the reason is that we don't understand very well the molecular, the pathways involved in this type of cancer, only the ones related to inherited forms. The advantage of GWAS is that we don't need any type of gene selection, so we don't have any bias towards specific genes. And using these GWAS approaches, we have been able to identify many genes involved in colorectal carcinoma. To apply GWAS means to use many cases and many controls. And for this we need consortiums. we are very happy that we have been working during all these years in organizing consortiums all over the world. In Spain, for instance, we have a very good consortium called EpiColumn that is integrated in an European consortium called COGENT. In this COGENT consortium, we have something like 40,000 samples, cases, and 40,000 controls. And with this study, we have been able to identify at least 15 genes that we know that they are very, very likely involved in colorectal cancer. And we have been able to identify 8% of the common variation. And that means that for the 34% of heritability, 5% can be explained by Mendelian forms. We have been able to identify 8% of the variability using this GWAS study, but there is still 21% of misheritability. For the time being, we don't have enough power to predict individual risk, but it could be useful perhaps to predict population risk. But at least we have been able to identify, with all these efforts and all this study, new targets and new pathways. We are still not able to stratify the disease, but we are going ahead quite quickly through a molecular stratification of the disease. And the problem, I strongly believe that is here, because these are the Mendelian forms. This is the copy number variation. It's quite interesting as well, but I can explain maybe 2% of the colorectal cancer. We are going to publish a paper in Nature Genetics next week with CMV variation in colorectal carcinoma, but only explain more or less 1% of the total variation. These are the GWAS studies, and the problem is the rare variation that is difficult to explore because it's not possible to do it with linkage analysis, and also impossible with GWAS that are always interrogating the hypothesis based in a common variation, common disease. I'm also very happy to see that there is a poster on exploring rare variation by the Professor Al-Katani group, exploring rare variation for breast cancer. I think we are doing a lot of efforts in this type of rare variation, and we have a project here to analyze this variation also in colorectal carcinoma. With all this information and regarding drug response, and with other ideas, we can use and we use in fact, we have developed panels of biomarkers to predict a response in treatments, and we can identify quite well the groups that people that they are going to respond, people that they are not likely to respond to the chemotherapy. But the problem is not response. The problem is to try to go ahead through a personalized medicine, and we need to find biomarkers for ADRs, for adversary drug reactions. But the main problem of the chemotherapy in colorectal carcinoma is that the response is not so bad, but the secondary effects are terrible. For instance, using the combinations that we use in first-line treatment, 25% of the people receiving FOLFIRI, they have neutropenia. 44% of the people receiving FOLFOX have neutropenia. And the same is true for many of the problems. Especially it's important with